Hi, and welcome to the Chapter 8b podcast on meiosis. This is Mr. Anderson. Before I get too far, MHSA AP student from YouTube asked me to give a shout out to Mr. White's class at Monteca High School in California, and so shout out. Hello from Montana, hello from my class, and welcome to YouTube. Before I get into the actual meat of the podcast, I wanted to share this picture with you because I think it's hilarious. This is from the 1600s. This is how they used to think babies were made, and so this is a sperm with a little baby inside of it. And you can even, the text is hilarious here. Man, the seed. Woman, the incubator. Now, nothing could be further from the truth. This is not how babies are made. And so my intent is to teach you how babies are made, eventually. Now, the function of meiosis is this, and a better way to think about that is the function of sex is to make sure that your offspring are different than you are, and they're different from each other. Because if we were all the same, then we all could easily be targeted by disease. And so one of these pictures is my father. So my biological father is pictured in A through I. And so your goal is to figure out which one it is. So there are probably clues you could pick up from my nose or my chin. But you can tell that we don't look exactly the same. And the reason we don't look exactly the same is meiosis. Meiosis, to understand how it works, you should also understand what a homologous structure is, or a homologous, chromosome is. And you may want to go back and look at podcast 8a if you haven't seen it yet. But a way to, I've kind of got some models that work good in class. And so the way to think about it is this. I've got a chromosome 1 from my dad, and I've got a chromosome 1 from my mom. And so these models kind of represent chromosomes. The beads then represent the genes that are found on the chromosomes, and where my fingers are represent the centromere, or the center. And so in both mitosis and meiosis, what happens is we make an exact duplicate of those chromosomes, and this is what you're familiar with when you think of a chromosome. In mitosis, what happens is they'll line up along the metaphase plate, and then each of those chromosomes will be pulled to either side, and so now you have two cells that each have the original amount of chromosomes in it. Now, meiosis is a little bit different than that, and so let me kind of go forward. In meiosis, what we're doing is we're actually mixing those chromosomes up, and so we get a new combination of those. And I'll show you that in just a second. So going back to the circle of life, last time in the first podcast we dealt with this, how we go from a zygote to an individual. How do we copy this one cell to the billions of cells that are in an organism? But today what I want to talk about is meiosis. How we go from those sex cells, so this would be in the testes and the ovaries of males and females, how we make eggs and sperm. And so how do we go from here all the way back to that fertilization? How do we complete the cycle? Now, meiosis, people have a tendency to get confused because there are bigger steps. In mitosis, I talked about IPMAT. In meiosis, we have IPMAT, PMAT, and so the only difference is that we don't have a second interphase. And so the first phase is going to look identical. And so after interphase, after we've copied the chromosomes, what happens is that those homologous chromosomes, since they've duplicated, will do something that doesn't happen in mitosis. In mitosis, they line up along that metaphase plate, and they split to either sides, or these split to either sides. But in meiosis, something happens that's really weird. And what will happen is the homologous chromosome, the one from dad and the ones from mom, will get next to each other, and they'll wrap around each other. And when they wrap around each other, they'll actually swap positions. And so instead of having chromosomes that were all red or all yellow, what you'll have is a combination of those. So then they'll line up in the middle, they'll meet in the middle, and then those will actually pull to the side. And so let me go to the keynote here. This is where that actually happens. So they're crossing over at this point. And since they're homologous, those chromosomes are the exact same length, they have the exact same genes, they're able to swap positions with each other. And so that's different than mitosis. That's the big difference right here. So next they'll line up along the middle, so you can see these. We have these are what are called tetrads, and you can think of why that's called a tetrad, because we have 4 of those chromatids hooked up at this point. The tetrads will actually separate. And so it's the same kind of thing where they meet in the middle in metaphase and they pull apart in anaphase. But now we go to Meiosis Two. And so what you'll get are those pulled apart chromosomes on either side. It'll then line up again at another metaphase plate. But the thing that's missing right here is interphase. And so since we don't copy the chromosomes again, since we don't go through an S phase where we're duplicating all the chromosomes, since there's no interphase right here, as we line up in the metaphase plate, pull apart, and then eventually go through telophase at the end, forming new nuclei, we've reduced the number of chromosomes. And so this is relatively simple because I'm just showing you one chromosome. But in humans, you can imagine the diversity we can get if you'd have 23 pairs of these all lined up, and eventually in Meiosis Two, those split. And so the cool thing about that crossing over is that it's created this chromosome that's different from this chromosome, which is different from here they are, these chromosomes as well. So they're all different. And so if you think about it, each of these 4 cells are going to be different than any other cells. And so each of those sperm and eggs are going to be totally different. And so this is to quote a song. I don't know if you saw this when you guys were growing up, but there used to be a show called Barney and Friends, and I didn't let my kids watch it. But Barney would sing this song, "You are special, you're the only one like you. There isn't another in the whole wide world who can do the things you do." Now, he wasn't a biologist, Barney the dinosaur, but he does talk about why we're special. And so there are 3 reasons why we're special. And if we talk about the first one, that's one that a lot of people don't understand. It's a big word, but essentially what it means is this. In independent assortment, there's a random chance that those chromosomes are going to line up either on this side or that side. Or they're going to be pulled in this direction or that direction, or this direction or that direction. And so who cares? You could think about it like that. Well, it's really important. Let's say there's a gene that causes, for example, Huntington's disease that's right there on this chromosome. You got it from one parent, but it's not over here. And so there's a 50-50 split. In other words, during independent assortment, it could go this way or it could go that way. If it goes this way, you are going to get a sperm or an egg that has that Huntington's disease, and you're going to get it. Or if it goes in the other way, you're not. And so it's really important that you understand independent assortment because it's going to make sense when you get to genetics. The second reason why you're special is to do that crossing over. If we didn't have crossing over, let's say this blue one came from your mom and this red one came from your dad, those chromosomes would just travel through time from generation to generation to generation, and they'd never mixed up. And so those genes, all of the genes on it would sit right next to each other. And so crossing over makes each of those chromosomes a new mix, a mix of your mom's and your dad's chromosomes. And then the last reason why you're special is the idea of random fertilization. And so in that one couple, so in that one mom and dad right here, they're going to produce, guys are going to produce trillions of sperm during their life. And so the odds of that one sperm fertilizing that one egg are pretty astronomical. So you really are special. Now sometimes things go wrong. And so the biggest thing to think about is what's called nondisjunction. And so nondisjunction is when instead of being pulled equally to the sides, some of them go too far in one direction or the other. What does that do? It creates cells that have too many or too few chromosomes. What usually happens, unless it's a sex chromosome, what usually happens is the zygote doesn't go anywhere. It can't advance any farther past that because it's just too many genes that are missing or too many extra genes. And so Down syndrome is an example of one that kind of can slip through. And so if you get 3 21st chromosomes, then you'll develop trisomy 21, or you develop Down syndrome. Down syndrome, there are a lot of characteristics that go around with that, but they can usually live a somewhat shorter, but a fairly normal life if you're Down syndrome. You can see in this graph that the chances of having a Down syndrome child go up drastically the older you get. The last thing then I want to talk about is review. And so if we look at differences between mitosis and meiosis, mitosis, 1 division, meiosis, 2 divisions, but meiosis also has that crossing over, and it also has a decrease in the number of chromosomes. And so in mitosis, if you look here, this cell looks exactly like that cell, but this cell makes 4 cells that are completely different and have half the chromosome of the parent cell. So I hope that